So we're looking at part 2 of membrane structure and cell membrane transport. And we'll be beginning with the first 2 learning objectives. So identify the 3 general factors that influence the rate at which a substance can be passively transported across a membrane, and then identify the 2 general factors that influence the rate of active transport. The second objective, which we'll also cover here as well, is to identify 4 factors affecting the permeability of membranes to molecules that cross by simple diffusion, and explain how and why each affects permeability. So just to recap what we've said so far, we looked at the structure of the membrane on Monday. All right, we talked about the phospholipid bilayer, and this is sort of setting the groundwork, right, the background for why some things will get through and other things won't. That phospholipid bilayer explains the semi-permeability of the membrane. The other thing we began looking at is the forces that start moving some particles across the membrane. So we looked at the membrane first, and then we talked about the forces that affect movement across the membrane. Today, we're going to talk about the factors, the environments, the conditions that influence the rate of movement across the membrane. And then we'll kind of put it all together and talk about how simple transport happens, and then how active and passive transport happens as well. So if we think about the rate of transport across the membrane, it's basically based on the concentration gradient. And so particles are going to be moving, as long as they can move through a membrane, they're going to be moving in one direction or the other, and they're going to be trying to get to a diffusional equilibrium. When they get to a diffusional equilibrium, there's going to be no more net transfer of molecules. All right. So if you look at the scenario here on the right, we're looking at 2 conditions. In both of these, we have a membrane that's separating both containers, and the membrane is semi-permeable to the solute that's dissolved in each of these solutions. In scenario A, we have equal particle distribution on either side. So side 1 has the same number of solutes dissolved as side 2. And I want to point out here that even though we don't have a gradient, we still see movement, what we call one-way flux of particles from one side to the other. So while there is no net accumulation, right, there's no net movement, we still see one-way movement of particles, but the overall concentration of both solutions will remain the same. In scenario 2, we have a gradient. So we have 2 moles of solutes dissolved on the left in side 1, and then 1 mole of solute dissolved in a concentration, the solution on side 2. So we have a gradient. We have higher concentration on side 1, lower concentration on side 2. So while we see one-way flux, there is a greater flux from side 1 to side 2. Therefore, there begins to be a net accumulation of particles from side 1 to side 2. So this is what we mean by particles moving to get to their diffusional equilibrium. All right. As long as we have a concentration gradient and we have a membrane that supports the movement of the solute, in other words, the membrane will allow the free movement of the solute in question, then we're going to always see the particles moving according to the gradient in order to achieve diffusional equilibrium, which is the scenario in A. Okay. So as we speak about passive transport, some of the characteristics: it is spontaneous, there is no cell energy required, it is a downhill type of movement. The types of passive transport that we see are simple diffusion, and simple diffusion means that the molecule or particle in question can go freely through the cell membrane. Facilitated diffusion, while it is still a type of passive transport in that there is no energy requirement and it's moving down the concentration gradient, facilitated diffusion, the particles here still require some type of help. They require a facilitated carrier or some type of transporter through which the molecules can move, because they cannot freely move through the membrane itself. All right. And so we'll talk about some of the properties of the membrane that will determine whether a molecule can freely go through or not. The other thing is that there is movement, passive movement by carriers and channels. So we can have diffusion of these molecules or particles, but they're going to require channels. The most classic example here are ions. And so ion channels are inserted in the membrane so that molecules can move. While it's still down their concentration gradient, ions are charged, and charged particles cannot go through the phospholipid bilayer, and so they're going to require some type of channel. Okay, water is another classic example. Water cannot move freely through that membrane. Remember, we said that water is polar. The center of the phospholipid bilayer is nonpolar, and so water is one of those things that cannot just go through the membrane, even when there is a concentration gradient that favors water moving into the cell. Right. So let us start with the factors that determine the rate of simple diffusion. So the characteristics of simple diffusion: no membrane proteins are needed. Transport happens freely through that phospholipid bilayer. So some of the factors that will determine the rate of simple diffusion: one, the magnitude of the driving force. Okay. Two, the membrane surface area that is available, and then obviously the membrane permeability. So when we speak about simple diffusion, we understand that the particle that is moving must be able to go freely through that phospholipid bilayer. In other words, one of the main characteristics is that it must be nonpolar. Okay, so let's illustrate a little scenario here to make that make a little bit more sense. So let's start by talking about the factors that affect the rate of simple diffusion. All right, so factors that affect the rate of simple diffusion. Okay, one is the magnitude of the driving force. And specifically the chemical driving force. Right, because we know that that is the driving force influenced by the concentration gradient. All right, so the magnitude of the chemical driving force, another way to say this is the degree of the concentration gradient, right? And that will certainly determine how fast, right? We're looking at the rate here, really important. So as we speak about the rate, it's how fast particles can move through the membrane, and the strength of the driving force will certainly influence that. The other thing is the membrane permeability, right? So the permeability of the membrane to the particle in question. Again, the particle in question must be able to go through that membrane for us to begin to think about influencing the rate of simple diffusion. If the particle can't get through the membrane, there is no simple diffusion, right? It must be able to move through that phospholipid bilayer. The third factor that will determine how fast simple diffusion occurs is the surface area that is available. All right, so the surface area available for transport. Actually, let's not say transport here. Let's say movement, because this is not being transported as such. This is moving freely through the membrane. So the surface area available for movement. All right. Okay, so let's look at a scenario to illustrate an example of this. So we've got three situations here. We've got a semi-permeable membrane in each of these. And we'll look at beaker A, beaker B, beaker C, and again, separated by the semi-permeable membrane. Now, the concentration gradient that we have in beaker A is 1 mole on the left and then 0 moles on the right, okay? So as you would expect, we've got a concentration gradient moving in this direction, right? Now, if we decrease the concentration gradient, let's say we had a smaller gradient. So we had 0.8 moles on the left, right? So let's do 0.8, but we had 0.5 moles on the right. So not very much of a difference. This is a smaller gradient. What we would see is that we now have a smaller driving force, right? So I'm going to illustrate that with a thinner arrow. Okay, so we have a stronger driving force because we have a bigger gradient or a bigger differential. We have a weaker driving force because we have a weaker gradient or a weaker a difference, right? In scenario 3, let's say we had no gradient. So we had 0.5 moles dissolved on the left, and we had 0.5 moles dissolved on the right as well. We have 0 gradient, right? What I want to illustrate here, however, is that we still see one-way flux from the left to the right and the right to the left, but we just don't see any net accumulation of particles. So in this scenario, scenario C, there is no concentration gradient. Okay, therefore the rate of movement equals 0, right? So the rate of simple diffusion equals 0. So there's no net movement. The other thing is that we can say that the particles here are at diffusional equilibrium, right? So the particles are at diffusional equilibrium. Okay, so there is no net movement, no net accumulation, and this is basically a concentration gradient of 0, therefore a rate of movement of 0. All right. Okay. Now, as we think about simple diffusion, the factors that affect simple diffusion. So we're going to kind of zoom in on simple diffusion. We'll talk about each of the factors that affect the rate, and then we'll talk about further factors that affect each of those individual factors. So make sure that you're staying with me as far as the level of simple diffusion that we're kind of looking at. So the factors that affect the rate are going to be the magnitude of the driving force. Therefore, if we look at this graph, on the y, we have the net flux, in other words, the net accumulation of particles. And then on the x, we have the concentration gradient and the change in the concentration gradient. So as the concentration gradient increases, the net flux of particles also increases. And that is a linear relationship, right? This is a linear, directly proportional relationship. So the degree with which the concentration gradient increases is the degree with which we see the net flux of particles increase as well. Okay. The other factor here, the membrane surface area, right? So if we increase the amount of area, right, the amount of membrane that is exposed for particles to move, we're certainly going to increase the rate of movement of those particles. And a good example of that is if we look at the alveoli in the lungs. So this is a histological image of the alveoli. On the left, this is in a perfectly healthy adult. So we can see these alveoli as we can appreciate, there's a lot of surface area, there's a lot of membrane here. And there's a lot of area for gas exchange. So oxygen and carbon dioxide can move freely across these alveoli because there's a lot of membrane or surface area exposed. In the image on the right, this is what would happen if we had emphysema, right? So emphysema, which typically results in collapse of the alveoli, so we lose that surface area. Notice how on the left, we can see that there's a lot of opportunity for gases to move because this is very well branched, right? So there's a lot of movement happening here. Because we lose some of the alveoli, because they collapse and expand on themselves, there's not as much free membrane surface area exposed. And so that would certainly influence how much gas exchange can occur in these patients. So this is a classic example of a factor that will determine the rate of movement across a membrane. If we don't have a lot of surface area exposed, it would just be intuitive to think we're going to see less movement across that membrane. Now, if we think about the other factors that affect the rate here, the third one is the membrane permeability. Now, if we take the membrane permeability and sort of look even closer at other factors that affect the permeability, the first one is the lipid solubility. Now, because the phospholipid bilayer is made of fats, right, because the center of that is nonpolar, in order for anything to get through that membrane, it must be lipid soluble. It must be nonpolar, it must be hydrophobic or lipophilic, and it must be able to get through that fatty acid center of the phospholipid layer. So the first factor that affects the membrane permeability is the lipid solubility of the substance or particle in question. The second is the size and shape of the diffusing particle. So if we have large, bulky particles like proteins, like glucose, for example, they're not going to be able to go through that membrane by simple diffusion. Even if there's a concentration gradient, if it can't permeate the membrane, there is no simple diffusion. So the size, the shape of the particle in question is definitely going to influence the membrane permeability, which is definitely going to influence the rate of simple diffusion. The other thing here is looking at the ability to go through that layer. So if it's a bulky particle, right, it's also not going to be able to go through that membrane as well. The third one is the temperature. So this is not always relevant in human physiology just because our bodies aim through homeostasis to maintain a relatively stable internal temperature. So there's not a whole lot of variety in our physiology, but if you look at external circumstances, if you increase the temperature, you're going to increase the rate of particle movement in general, and so you're definitely going to influence the rate of movement across a membrane. Okay, the last factor that influences the membrane permeability is the thickness of the membrane, right? The thickness of the membrane. So if we have a very thick membrane, we're going to lose the ability or decrease the ability for particles to move through at a faster rate. This is why in areas like the alveoli of the lungs, in areas like certain membranes, if you think about areas where we need to have gas exchange occurring, the membrane has to be very thin. Going back to that alveoli, that example that we looked at before, notice how that cell layer has to be one cell layer thick in order to accommodate the free movement of gases like oxygen and carbon dioxide. So the thickness of the membrane is certainly going to influence the permeability, which is definitely going to affect the rate of simple diffusion. All right, and this kind of just makes sense naturally. If we have more stuff in the way, we're going to decrease the rate. It's going to kind of slow down the rate of movement. If we look at the illustration here on the right, if we think about the concentration gradient again on the X, the net flux on the Y, if we have a low membrane permeability, so if we have factors that decrease the permeability, so let's say we have a thicker membrane, let's say we have less surface area available, we're going to see a lower rate of movement. And we're going to see, even if we have a high concentration gradient, the rate of movement is still going to be decreased. On the other hand, if we have a high membrane permeability, so we have a particle that is non-charged, it's lipid soluble, it's small, it is, we also have a thin membrane, we have all the right factors, all the right conditions that will increase membrane permeability, we're going to see that even if we had the same concentration gradient, we now have a higher net flux, we now have more movement simply because we have a higher membrane permeability. All right, so the membrane permeability will certainly influence the movement of particles, the rate of simple diffusion, even when the concentration gradient is the exact same.